Good afternoon, dear participants. Welcome to day 6, session 3. Today, we have with us Dr. Ketan V. Hatware, sir. He is a senior lecturer at School of Pharmacy, IMU University, Kuala Lumpur, Malaysia. He is going to deliver a talk on integrating organ on chip technology into education and research. Regarding his educational qualification, Sir is PhD in Pharmaceutical Sciences Pharmacology from Andhra University in 2017. He has completed Masters in Pharmacy in specialization of Pharmacology subject in 2008 from Andhra University, B Pharm in 2006 from Sant Gadge Baba Amravati University. He has also completed his Masters in Yoga Shastra in 2022 from Kavi Kulguru Kalidas Sanskrit University. He has also completed Yoga Teacher Diploma in 2020 from Yeshwantarao Chavan, Maharashtra Open University. Regarding his work experience, he is currently working as a senior lecturer in IMU University, Malaysia, School of Pharmacy at Life Sciences Department. Awards and achievements for Dr. Ketan. He has a research funding seed grant in 2018 and 2019 on the title Effect of Cow Urine Residue and its Fraction on Sperm Quality and Quantity of Diabetic Male Vista Rats. He has supervised master's research, particularly research on anti-cancer activity, memory deficit, antioxidant activity, anti-hypolipidemic activity. Sir is a member of Indian Pharmaceutical Association since 2019. Sir is also the member of APTI, that is Association of Pharmaceutical Teachers of India from 2019. Regarding his publications, Sir has published different research articles into the reputed journals like Current Cancer Therapy Reviews in 2025, Frontiers in Medical Technology 2025, Current Drug Therapy 2024, Current Diabetes Reviews 2024, Helion 2024. Sir has very good Google Scholar profile. He has overall 766 citations with H index of 13 and I-10 index of 15. So with this brief introduction, I would like to request Dr. Ketan Hatware sir to initiate the session for the today's audience.
Yeah, thank you. Thank you very much for the introduction. And I would like to thank Gokhale Education Society and Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, Nashik, for giving me this opportunity to deliver the talk on a particular topic, that is integrating organ on chip technology into education and research. These are the contents of my today's talk. New drug development, drug toxicity studies, and long-term adverse effects of drugs are the major concerns in today's era. That we have found that many of the drugs, it will be effective in preclinical studies, but later on it will fails to come into the market because of the toxicities and certain complications. Thus, there is a requirement of advanced technologies to fulfill all the criteria for rational drug utilization and the patient care. So definitely there will be a need of the novel technologies and advancement for the new drug discovery process, which will enhance the speed as well as effectiveness of the new drug. With this background, I would like to just talk on organ on chip technology. The concept of organ on chip technology was significantly elaborated by Dr. Donald Inberg and his team at Wyss Institute of Biologically Inspired Engineering at Harvard University in 2010. The key contribution of his team and himself was they have developed the lung on chip was the first successful organ-on-chip system. They have demonstrated how the microfluidic device could replicate key functions of the human lung. Generally, in case of in vitro and in vivo studies, that there will be a certain limitation, some lacunas will be there. In vitro studies may not resemble with the natural environment which is there in the human being or even in the animal. But the organ-on-chip system is such a system where the microenvironment will be developed which resembles to the natural microenvironment which is present in the cell, tissue, and organ. The global impact of such organ-on-chip technology is the technology has since been adopted by pharmaceutical companies, research institutions, and regulatory agencies like FDA, NIH, and European Medicine Agency for drug testing and disease modeling. So drug testing will become more authenticated and more effective, and definitely to understand the basic pathogenesis and advanced macromolecular, micromolecular level pathogenesis in the different kind of diseases and disorder, it is possible with the organ-on-chip technology. There are certain startups like Emulate are commercializing the organ-on-chip system for biomedical applications. So that in the future, further slides, I am going to explain that. Basically, what is the organ-on-chip? That organ-on-chip are tiny devices designed to replicate the microenvironment and physiological conditions, which resembles to the natural microenvironment within the body. It is used to provide more realistic responses. The chips are often made from synthetic transparent materials and contain minute hollow tubes. As our body, as our organs, as our biological systems are having the blood vessels, extravascular fluid, intravascular fluid, then the matrix will be there. Along with that, number of proteins, receptors, entire microenvironment which should be resemble to the natural microenvironment. And because of that, the exact effectiveness or the exact effect of the xenop can be identified with the help of organ-on-chip. This organ-on-chip is used to enable more precise simulation of the natural cellular microenvironment, because nowadays the simulation studies, the docking studies, and etc. are very, very useful in the new drug discovery. Those type of advanced tools can be utilized with the help of artificial intelligence and machine learning, so that the unnecessary wastage of time or sometimes what will happen if your procedure is not appropriate, there will be a loss of monetary aspect also there. So to enhance the effectiveness and to get the precise simulation, the organ-on-chip is one of the important tool which can be utilized. The organ-on-chip is also having a transparency and integration of biosensors, which can provide more detailed and accurate insights into the molecular and cellular activities. So you can find in the picture that initially the lungs on the chip was developed, where in the diagram you can observe that there will be a lung cells, there will be a the microtubules which are having the process where the inspiration and expiration will be taken place, how the blood flow is going on, how the drugs are going to be absorbed, how the capillary cells are present in the lungs itself. So likewise, a microenvironment will be developed which can be helpful to analyze the therapeutic as well as some undesirable type of effect of the test compound, test chemicals, and etc. Recently, you can find that in the coming in few years, in the last few decades, you can find that rapid growth of this technology in biomedical research. You can find that in 2000 there were negligible publications and research which was done on the organ-on-chip technology. But now, in last year, in 2024, 791 publications in PubMed, it is reflecting that the researchers are focusing on this novel technology. In drug development and patient care, the FDA has officially accepted the first submission of the organ-on-a-chip technology in 2024 under its innovative science and technology approaches for new drugs pilot program. There they have accepted initially the liver-on-chip. The liver-on-chip was the one of the system which was used for the assessment of risk of drug-induced liver toxicity. Organ on chip is a microfluidic device that mimics the structure and function of human organs, so which resemble to how the organs are behaving, how the physiological systems are coordinating with each other. So those kinds of environment will be given with the microfluidic device, where the extravascular, intravascular fluid will be there, blood flow, blood perfusion will be there, and it resembles to the similar kind of environment. These chips are developed using microfabrication techniques that replicate the microenvironment of a tissue, which resemble as the exact tissue also. This allows researchers to study disease mechanisms, drug responses, and toxicology in a more human-relevant way than the traditional in vitro and in vivo models. So traditional in vitro, in vivo models are having a lot of concerns. At the same time, there were limitations, and we cannot resemble the effect same as that of the human or biological system. But in case of organ on chip, the assessment will be done with the pesticide, nanoparticles, drugs, chemicals, environmental factor, and any kind of xenobe, where it will be initiated with the particular concept, then it is formed in a three-dimensional culture, then it is compounded in a particular microfabricated material, which are number of hydrogels or polymers can be incorporated. Along with that, there will be a special feature that is a biosensor. Integrating the organ on chip technology in education enhances the students' understanding of human physiology, pharmacokinetics, and the drug interactions in a dynamic and interactive way. So this is possible with the exactly how the physiological system is working, how the drug is moving within the body, like pharmacokinetics, that real-time effect of the drug when it is enter in the body, that how it is absorbing, how it is distributing, how it is metabolizing with the help of various enzymes, and how it is going to be eliminated from the body. With that, how the affection will be there towards a particular protein, which will be considered as a receptors, and how the cell transduction mechanisms will be going on, that can be predicted with the help of organ on chip technology. So there are three major aspects in the organ on chip technology, where microfluidics, 3D cell culture, and biophysical signals. Organ on chips use small scale fluid channels to circulate nutrients, drugs, and other molecules simulating the blood flow. As when we are consuming a drug, how the nutrients are moving, how the other internal endogenous materials or neurotransmitters or biochemicals are moving, how the secretion of the cytokines will take place based upon the situation. Other molecules are moving along with the blood. The similar kind of microenvironment will be provided by the organ on chip technology. Even 3D cell culture, which is the advanced stage than that of our traditional cell culture and 2D cell culture, that the cells in an organ on chip grow in a more natural and three-dimensional environment, so which will expose to entire environment which is present in the natural organs and natural biological system. The biophysical signals like organ on chip technology integrate mechanical forces like shear stress, where the blood flow is moving, how the resistance will develop, how particular perfusion is going on, it will resemble in the same technology. And the stretching, bending, or you can say that contraction and relaxation type of process also can be incorporated in the organ on chip technology, like breathing or respiration. In case of the lung on chip technology, they already use for the assessment of the effectiveness of certain drug used for the respiratory complications. It replicates the real physiological condition, and that's why it is giving more profound and more authenticated results when compared to the traditional techniques. Here you can find that there are a number of examples of organ on chip models, like lung on chip, heart on a chip, liver on a chip, kidney on a chip, or brain on a chip. So all these are the various models where the lungs on a chip can be useful for exploring the lung function, including the breathing motion and the gas exchange, that how the oxygen and carbon dioxide will be exchanged in the similar way, that organ on chip also giving the similar kind of feature. Heart on a chip, where exactly the assessment of cardiac contractility will be possible, and it will be helpful to study the heart diseases, how it is occurring, what is the pathogenesis, and how the drugs are affecting that particular system that we can identify. The liver on chip is specifically used for the toxicity testing, that number of drugs which are having the hepatotoxicity that can be analyzed, that can be predicted, and even the liver metabolism can be analyzed with the help of liver on chip. Kidney on chip helps to analyze kidney filtration and drug clearance. Eventually, the brain on chip is one of the model used for assessment of neurological diseases and drug penetration through the blood-brain barrier. We know very well that the blood-brain barrier is one of the important barrier, so that the number of xenobes will be prevented to enter into the brain or central nervous system. But whenever there are certain diseases like Alzheimer's disease or related complication, there the drug Parkinson disease, where the drug has to cross the blood-brain barrier and produce the effect in the central nervous system. In those circumstances, to analyze exactly that whether the test compound or our drug compound is crossing the blood-brain barrier or not, that can be easily identified with such type of brain-on-chip model or blood-brain on chip model. So you can find in the image that number of chip models have been developed by various researchers and companies that it will be useful for different applications, like blood-brain barrier to identify the permeability towards the CNS, the lung on chip where the respiratory rate as well as the drug absorption through the inhalation and all can be analyzed, the gut on chip, along with that the liver on chip for the toxicity studies, the kidney on chip for the elimination and assessment of the T-half of the particular drug, and the heart on chip for assessment of the cardiac physiological condition and the effectiveness of drug on the cardiac system. Above all, there is body on chip. So initially they have developed the single organ on a particular chip so that the exact impact of drug or test compound on a single organ can be analyzed. But nowadays, the advancement is going on that how exactly the multiple organs are present in the body. Similarly, multiple organ chip will be developed so that the exactly how the drug is, when drug is entering in the body, how it is moving across the body and how the drug is binding to the specific target and showing their effect, that can be analyzed with the particular body on chip. It is the ongoing research in this particular area. The organ on chip research has been one of the most fruitful areas of the entire field of tissue engineering. It is a combination, and you can say that it is a collaborative work of the pharmacologist and the biomedical engineer and the experts from the artificial intelligence and machine learning, so where the biosensors can be utilized. So that's why this field is growing day by day, where the journey of organ on chip will be described with the following steps, like the initial idea or the concept of the idea, then development of design, then fabrication and materials, and incorporation of biosensor in the particular including the biomaterial, the biology consideration where the cell sources and biomaterials should be taken, like whether it is for generalized treatment and management of disease, or it is for the personalized treatment where from the patient itself the cells should be taken, incorporated in the organ on chip, and then the medication should be tested for the same patient as a for the getting personalized medication result. The cell injection and tissue assembly process, which will be done with the three-dimensional structure with this advanced cell culture technique, and the development of assay is very important thing for the assessment of content of drug, concentration of drug in that particular region where we are going to incorporate the drug. The functional validation of the assay process should be done, and finally there will be a application of the developed organ on chip with various applications in pharma industry, clinical research, and mechanistic biomedical studies. You can find the flow chart here that how the idea is going to be developed, how the cell sources will be decided, the selection of the biomaterials and fabrication of that biomaterials, along with that incorporation of sensor, and then there will be a development of the assay and the validation of that assay, which will be helpful for these three major applications. Coming to the materials, that generally there are four major type of materials like polymers and use of advanced cell culture techniques in the organ on chip. You can find here that PMMA or PDMS and OSTE, these are the common polymer used for the organ on chip technology development, and the advanced cell culture techniques which will be used, which will be associated with the micro pumping system, where the cells will be incorporated, and it is in between the upper compartment and the lower compartment where the supply of microfluidic channel will be there, which is giving the tunnel-like structure, small tunnel-like structure, blood vessel-like structure, and the matrix which will be present, which will be generated by the polymer as well as the hydrogels. It will resemble with your extravascular matrix and intracellular fluid also. The most commonly the polymers are the material of choice, such as polydimethyl siloxane, that is abbreviated as PDMS, and it is more compatible with most of the cell type, and that's why generally it will be utilized by the researcher to develop the organ on chip. It is not that much costly, it is inexpensive and easy to handle, and having more stability also. Other materials also can use, like in case of micro physiological system, which include the polymethyl methacrylate, the polycarbonate, then of stoichiometry thylene, and other materials also can be used. Even glass can be used for the development of organ on chip. Along with that, the 3D matrices, which will be developed with the help of hydrogels and other type of materials, impart the feature of native extracellular matrix, which is with the biochemical composition, biocompatibility, and the properties including the porosity, elasticity, stiffness, and degradability, as well as permeability. All the features which the cell, cellular membrane, the organ, biological system will have, that similar kind of structure will be developed in an organ on chip, so that exact impact of the drug, exact effect of the drug can be analyzed. United States Food and Drug Administration considered that the microphysiological system as an advanced cell culture platform for in vitro modeling of functional elements of a specific human or animal tissue or the organ system. So even FDA also has given the approval that we can use the organ on chip technology as an advanced in vitro modeling, which will replace the traditional techniques which we are using for the in vitro toxicity and other type of assessment. The major role which will be played by the biosensor and detectors. We know very well that the biomarkers in our body are one of the complex criteria, and assessment of biomarkers is very, very essential for the early diagnosis. At the same time, that whenever we are giving the treatment, the change in the levels of biomarkers needs to be analyzed perfectly, so that we can assume that given treatment is appropriate, or the given treatment fails to our objectives, or the given treatment is causing some undesirable effect, etc. So for that purpose, there are the biosensors which can be incorporated in the organ on chip technology, where electrochemical biosensors for the biomarker detection, specifically for the cardiac marker like troponin and CKMB during the cardiac complication like cardiac arrhythmia, cardiac myocardial infarction, and in case of liver functioning test, what are the markers are there, like SGOT, SGPT, ALT, ASP, all the liver function test biomarkers can be analyzed with the help of a biosensor. The drug detection markers like acetaminophen in hepatocyte for the liver on the chip itself, we can identify it, like we know very well that acetaminophen is one of the known hepatotoxic agent, and we have to analyze the level or the concentration of acetaminophen in the liver tissues, so that if it is crossing the limit, then at that time it may cause the hepatotoxicity. For that, the early detection with the biosensor is possible, and with that, in case of chronic treatment for rheumatoid arthritis or chronic pain, or in case of regular use of the acetaminophen, that can be controlled by identifying the concentration of a particular drug with the biosensor. Along with that, the biosensors are useful for assessment of the concentration of pro-inflammatory marker like TNF-alpha and interleukins, along with that other cytokines also. So not only pro-inflammatory marker, even endogenous anti-inflammatory marker can be analyzed or detected by the biosensor, which will be helpful for our research for assessment of the therapeutic effectiveness of our given substance. You can find here that there are various biosensors which can be incorporated in single organ chip as well as multi-organ chip, which is connected with the electrode and the imaging system, which is connected with the monitor, so that it is directly reflecting how the changes which will be going on in the presence of a particular drug or the test substance. So here you can find that how the development of organ-on-chip technology is useful and how it can be developed. So you can find that from the human being or from the animal, the cell can be isolated. So isolated cells, then it will be cultured. Later on, the 3D culture, or you can say that hydrogel encapsulation will be done, which is forming an advanced form of culture. Then the utilization of 3D printing also very effective and very essential part of the development of organ-on-chip technology, and followed by organ-on-chip, which will be developed with the help of biosensors and all, which include the epithelial cells, the membrane, then endothelial cells, the microtubules, blood vessel-like structure, and etc. with the biomaterial. And the biosensor will be available, which can detect the drug, and the biomarkers also, which will be highly sensitive with any kind of stimuli. As our body is sensitive for different kind of stimuli, that organ-on-chip also have the resemblance for the sensitivity, and it is going to show the changes with respect to the stimuli. That will be observed on the monitor, which is connected with the organ-on-chip. There are a number of researchers who are working on the organ-on-chip technology in a very advanced manner, and the advanced technology is going to be emerged. Like organ-on-chip technology is integrating the cutting-edge innovation in microengineering, biomaterials, and computational modeling to enhance its application in a drug discovery, disease modeling, and personalized medicine. These are the very important areas for the research, because the new drug discovery is in need of time. Disease modeling to understand exactly how the disease is going to be occur, how we can prevent the disease, and how best we can treat the disease with the help of particular drug that can be identified with the disease modeling. And personalized medicine, where we found that many of the people they are highly susceptible or highly sensitive for a kind of drug. So those kind of drugs should be avoided, and for that personalized medicine, so patient specific medicine is most important criteria to achieve the best therapeutic outcome and to minimize unwanted allergic and adverse effects and toxicological effect in the patient. So for that, the organ-on-chip technology is one of the important tool which can be very useful. Coming to the applications, the major applications of organ-on-chip technology, that is drug testing and drug development, disease modeling, and personalized medicine. Just now I told that these personalized medicine and disease modeling is one of the important criteria for the better management of disease as well as disorder. In case of drug testing and drug development, it provides a better prediction of human responses to the drug, reducing the reliance on animal models. We know very well that there are stringent rules and regulation, and the authorities are there, ethical committees will be there, that they will reduce, they require or they want to reduce the usage of animals. And it is ethical also, because unnecessarily if we are using the animal for the toxicological study or to get the lethal dose or to identify the LD50 and etc. So unnecessarily we are going to sacrifice the animal. But if we are using the advanced technology, that software-based modeling and this organ-on-chip technology, then it will reduce the reliance on the animal and it will fulfill the criteria for the authorities and the ethical committees also. Along with that, it will give the responses and the results which resembles with the human being. We found that many of the drugs which pass the toxicity test in the preclinical studies and in the clinical trial, it fails because of the adverse effect, long-term adverse effect, or some sort of unwanted toxicological effects also. In those circumstances, that sponsor has to bear a lot of monetary loss and time also going to be wasted. So to avoid such type of thing in our drug development, if we include the organ on chip technology, it will give the fast and perfect type of results. Whether the drug can move ahead for the clinical trials or not, that we can decide earlier. Disease modeling, where it helps the study conditions like cancer, Alzheimer's disease, and many complicated disorders and diseases also, where exactly which biomarkers are responsible or releasing during the disease condition, or which kind of etiological factors are responsible for that, that we can identify. Along with that, in case of personalized medicine, we can use the patient-derived cell to test the drug responses for tailored treatment. So it will be only for that particular patient only, and it will be more useful for that patient. Here you can find the further application that biological mechanism can be identified using the lung on chip. The disease modeling, again, it can be done with the help of lung on chip, where a pulmonary edema and lung cancer can be identified. Again, the ability of accomplish a new drug discovery and toxicity test with the liver on the chip. Regenerative medicine such as neuron recovery and stem cell differentiation, specifically in case of neurodegenerative disorders, this organ on chip, where the regenerative medicines, whether it is effective or not, that can be identified. The drug potential in radiobiology. The great potential in radiobiology studies is developed using the bone marrow on chip and the gut on the chip, where the radiological aspect and the toxicities as well as impact of some radiological substances on the bone marrow as well as the gut wall, that can be identified with the help of this organ on chip technology. In case of drug development and safety testing, that liver on chip, already I told you that pharmaceutical companies, to test the drug-induced liver injury, which is the major cause for the drug failures in the clinical trial. Because liver and kidney, these are the important organs which are maximum coming in contact with the drug after its administration. Because the blood perfusion as well as the size of organ depends upon their size, that drug distribution also takes place. And we know very well, most of the drugs are metabolized in the liver and eliminated through the kidney. That's why the organ on chip with the liver and the kidney will give you the clear-cut idea that how the pharmaceutical product are going to be metabolized, whether at which rate it is going to be metabolized, and how long it will take time to eliminate from the body. Along with that, the drug and drug metabolites, if they are causing some toxicity to the liver and the kidney, that can be identified. So here there is an example that AstraZeneca and Emulate are the company in a collaboration. They have developed the organ on chip to predict the toxicities responses of the human tissues, specifically for the liver and kidney, against the various kind of drug and the test components. Improving the drug efficacy. In case of heart on chip, we have seen that it is used to study the effects of cardiovascular drug and prevent the adverse reaction like arrhythmia. We have found that many of the antiarrhythmic drugs, they themselves can cause the cardiac arrhythmia, and because of that reason, that exactly how much extent the drug should be taken, how long drug should be present in contact with the myocardium and the heart tissue, that can be identified with the help of organ on chip. Lung on chip will give the particular effect of drug which can be used for the respiratory complication, and how the drug is going to be delivered after the inhalation use or inhalation route of administration. It can be identified for the prolonged conditions or chronic conditions like asthma and chronic obstructive pulmonary disorders also, where the effectiveness of drug can be identified using the organ on chip technology. It will helpful for improving the drug efficacy and to identify even the toxicity outcomes also. Coming to the educational benefits of organ on chip, definitely any novel technology or new techniques which will be used in the research and the pharmaceutical field, it will be helpful for the budding pharmacists for their educational purpose also. The hands-on learning, where the student can observe real-time effects of micro-engineered tissue, that exactly how the drug is acting, how the drug is having the impact on that particular microenvironment, that can be identified, that can be observed using this organ on chip technology. And for that, the particular softwares and experimentation should be done, and it should be included in the curriculum also, so that the student can identify how the drug will acting on the different tissues, organs, cells, and the biological system. The bridging theory and application, that it is the link between the classroom knowledge and the cutting edge experimental techniques, because we know very well that the knowledge of the classroom, which will be giving the traditional aspect, and when we are going at the workplace, at that time advanced tools, advanced techniques will be utilized. So incorporation of the advanced technology like organ on chip in the drug assessment, drug safety, and the development of personalized medication, it will be a bridge between the classroom knowledge and the cutting edge experimental techniques. Along with that, ethical alternative. Definitely, as we have seen that the toxicity studies require the ethical approval, where the animal ethics committee has to give the approval for the testing, and they try to reduce the reliance on the animal testing, and it should be aligned with the 3 R's like replacement, reduction, and refinement. So in that case, the technology like organ on chip is helpful, which can be replace the animals and getting the similar kind of effect, even more advanced and more authenticated report for getting the results, getting the report which can be useful for further clinical trials and further process of the drug development. These principles are very useful in case of the research, so that the replacement of animal will give you the more authenticated responses of a drug on a biological system. Interdisciplinary exposure. Definitely, the study on the organ on chip encourages the collaboration between the pharmacology, biomedical engineering, and computational modeling fields. So 3 and 4 different domains, including artificial intelligence and machine learning, also need to be collaborated with this technology, so that the different domains and expertise from the different domain, when come together, the outcome will be very good and very effective. Applications in the pharmacology research, that already we have discussed that there will be toxicity studies as well as effect of the drug and efficacy studies can be done with the organ on chip technology. Along with that, high throughput screening, which allows the researchers to test multiple drug compounds efficiently at once. For example, if one of the person is having multiple complications, multiple disorders and all, and he is on polypharmacy, at that time, how the drug interaction takes place, how multiple drugs acting on the different organs at once, that we can identify with the help of organ on chip technology, specifically with multiple organ on chip technology. The predictive toxicology, that we can predict initially that whether our test compound, with the help of docking study and the organ on chip technology, that the models such as liver on chip help to assess the drug toxicity and metabolism in human being, or which resembles to the human-like condition. So liver on chip will resembles the similar type of metabolic rate and the metabolic aspect, the assessment of enzyme induction, enzyme inhibition by other biomarkers, or by other concurrently administered drug or etaples. That kind of environment we can create with the help of organ on chip technology. While disease modeling, so specifically like cancer-like model or Alzheimer's disease-like model we can develop. We know very well when we are going to test the drug substances on the animal models, and we are going to develop the cancer model in animal. So it is somehow we are causing some cancerous cell or inducing cancerous cell in the animal, or using some chemical agents and all. And it may not resemble with the human being, or the cancer which is developed in the human being. In those circumstances, if we are using the organ on chip like technology, it will give the resemblance which exactly as that of the human being, and more effective drug can be identified for the same kind of system. So that's why disease modeling, to identify the pathogenesis, exact model can be developed as that of the natural, or you can say that human beings are going to suffer from cancer or some of the disorders like Alzheimer's disease. Even dementia-like condition is very difficult to create in animal, but this new technology can helpful to create the environment which resemble to the central nervous system in the human being. So that's why it can be useful for neurodegenerative disorders, cancer-like condition, and related complications also. Specifically, along with the pharmacokinetics, the pharmacodynamic effect can be analyzed with the help of organ-on-chip technology, where the real-time drug absorption, distribution, metabolism, and excretion pattern can be identified. And that's why the organ-on-chips can mimic the organ-specific drug responses. So how the drug is entering, where it is moving, whether it is moving to the liver, how long it will stay in the liver, or whether it is moving to kidney, how much time it is taking to eliminate from the body irreversibly. So all these things can be identified. Even how the metabolism takes place and what is the feature of the metabolite that can be identified with the help of organ-on-chip technology. Whether the drug is effective, whether the drug is forming a toxic metabolite. So those kinds of things can be identified. Even multi-organ chip for the systemic effect is much more useful, where a human on a chip model connect the multiple organ system to study the drug interaction and side effect for more efficient manner. As if we are taking only one drug, we know that the drug is having so and so structure and having the affinity toward so and so parameters, so and so targets and all. And based upon that only it can show the effect. But once we are taking multiple drugs, at that time there will be a drug interaction. And chances are there will be formation of complexes. That complex may bind to the different targets also. Sometimes it may not absorb, or sometimes it will not metabolize. So that kind of alteration can be identified with the help of multiple organ chip technology, and which will be helpful to find out the drug interaction and final outcome of the multiple drug utilization also. So use of 3D bioprinting and stem cell integration is one of the advanced research which is going on in the organ on chip technology. Bioprinted organs on chip, which will be used the patient derived stem cell to create more physiologically accurate tissue. And because of that reason, we can identify the exact target and we can identify the exact problem upon administration of drug, how it is taken place in case of the body or in case of human being. So that resemblance in the organ on chip technology will helpful to get the exact effect of the drug and the test compound for the particular patient himself. Induced pluripotent stem cells, it is also one of the important aspect of advanced research in the organ on chip technology, where it enables the personalized organ chip for the precision medicine. So always when we are using a drug for the long-term purpose or for the chronic disease, at that time precise effect should be there. And with the precise effect, the more betterment or more therapeutic effectiveness can be achieved, which will be helpful to improve the quality of life of a patient. So that's why these advancements are very, very useful for the personalized medication. Similarly, extravascular matrix engineering. So as initially at the time of fabrication, I mentioned that the use of hydrogels and the biomaterials, those combination of biomaterials, biosensor and hydrogel is helpful to maintain the similar type of matrix like extracellular matrix, which will be enhancing the cell attachment and the function in the chip, so that if the drug is having affinity towards the extracellular matrix or extracellular proteins or extracellular component, that also can be identified, and with that we can get the exact outcome of the drug and drug pharmacological action on a particular organ. Definitely there will be a role of artificial intelligence and computational modeling, which is very, very important, and it is a need of time. AI-powered drug screening is useful for assessment of the predictions of toxicities and the therapeutic outcome also. AI algorithms analyze organ-on-chip data for rapid drug discovery. So definitely it will be helpful to reduce the time of the drug discovery. So in the traditionally that a lot of time used to be taken by the drug to come into the market, but now with the advanced tools and the docking studies as well as AI powered drug screening methods, that the duration of the drug discovery is going to be reduces, and the drug, new molecules, new drugs, advanced drug, highly effective drug can come into the market as soon as possible. Digital twins, where the virtual patient models used for organ-on-chip data to predict the real-world drug responses, that how the drug is going to be responding, or how whether the body is tolerating the drug dose, or long-term effect of the drug, how it is going to reflect, whether it is toxic or whether it is giving the therapeutic effect, or whether it is curing the disease and disorder in best time period and all, that can be identified with the virtual patient model developed with the help of organ on chip technology. Machine learning for toxicity prediction, where the AI processes the organ on chip experiment to predict the adverse drug outcome also. So we generally, when we are going for the traditional techniques for the assessment of toxicity as well as adverse drug effect, adverse drug reaction, we are going for acute toxicity, subacute toxicity, and the long-term toxicity. But prolonged use of drug, how it is going to cause toxicity or some adverse effect, that we cannot identify, because it will take a lot of time. And that's why if the technology like organ on chip will give you the prediction that long-term use of drug leading to which kind of adverse drug effect, that will be more useful to identify the prolonged drug adverse effect and adverse outcome, which will be helpful for the new drug discovery. Along with that, in case of infectious diseases and immunology research, where the immune system on chip is one of the advanced tool which can be used, which stimulates the immune responses to the pathogens and the vaccines. How the pathogens are entering in the system, how it is going to be divided, and how it is going to develop the resistance toward the medicament, that can be identified. Along with that, the effectiveness of vaccine for the prevention of the infectious diseases, that can be also identified, and the treatment, that once the drug become resistant, if new drug need to be identified for the effectiveness, therapeutic effectiveness in that same kind of infection, then the immune system organ on chip is much more useful, where multiple drug can be utilized, and you can identify the drug combination or individual drug, how it is effective, and how which drug can be useful for the drug resistance infections and all, specifically for tuberculosis and related complications, related infections also. For COVID-19 research, where the lung on chip was used for the infection caused by SARS-CoV-2 infection, where the antiviral treatment or antiviral drugs was analyzed for the on the organ on chip technology. That chip was the lung on chip, which was given the effectiveness of several antiviral drugs for their treatment. Microbiome on the chip investigates the impact of gut bacteria on human health and the diseases. So all these are the advanced things which are under development, which are under trials, where these kinds of organ on chip useful for the new drug discovery and identify the exact impact of the drug on the human body. Along with that, the space and extreme environmental research, where we know very well that based upon the changing environmental condition and in the space research also, the NASA's organ on chip studies, where organ on chip are used in space research to understand aging, muscle atrophy, and radiation effects in the microgravity and etc. Where once the astronauts are going to the space, definitely the food is going to be changed, the routine is going to be changed, the spatial environment will be totally different from the Earth. So in those circumstances, that microgravity impact and all, which will be much more higher on those people. So because of that reason, the NASA has developed the organ on chip for such studies, that what is the impact of the microgravity on the organs and the different physiological function that can be identified. This is the advanced application in the research with respect to the organ on chip technology. Along with that, toxicology in hazardous environment. For example, a person who is working in the toxic environment, in the industry or in the environment where some fumes are there, some chemicals are there, always the person has coming in contact with such type of hazardous chemicals and all. To identify that, how long that person should work efficiently, and if he is going to affected with these hazardous chemical, that can be identified, that can be predicted initially with the organ on chip technology. So in that case, we can predict such type of adverse outcome in case of environment and the special things and etc. So that the toxins or the impact of microgravity can be analyzed using the organ on chip technology. Coming to the future advancement, where the artificial intelligence and machine learning integration in the organ on chip technology, where AI driven analysis and robotic platforms of drug testing of organ on chip technology data cloud accelerate the drug discovery. Because in case of AI driven analysis, where multiple data will be there, big data set will be there, and the robotic platform where this exact process, for example, the 3D installation of the cell culture and all will be done with the help of robotics and advanced technologies, where the data can be useful for the predictions of multiple utilization of drug and the prediction of the outcome, adverse outcome of the drug on the multiple organs. So that's why the artificial intelligence and machine learning need to be integrated in the organ on chip technology, so that the more profound data and more useful data we can obtain. Similarly, 3D bioprinting and stem cell technology further enhances the organ on chip technology complexity and realism, so which resembles to the human being, or which resembles to the natural biological system, so that exact outcome can be identified and can be analyzed. Though the number of applications are there and advanced things are incorporated in organ on chip technology, there are certain limitations with this particular tool. That is standardization and reproducibility. Definitely, the standardization and reproducibility of such organ on chip technology is very difficult in current conditions, which need a uniform protocol for the chip design and the data interpretation. Because company to company, researcher to researcher, it is going to vary. But there is no such a gold standard. And that's why the standardization and reproducibility is one of the major concerns, and it is one of the limitation. But it can be overcome with advanced research and the focus on this particular technology. Then scalability and manufacturing. Definitely, mass production of organ on chip technology remains a challenge, because it requires the advanced tools, advanced techniques, the expertise from the different field. So it is quite difficult and expensive affair also. And because of that reason, the chances are there that mass production of the organ on chip technology is one of the major challenge in the present era. Regulatory approval. Though the FDA and European medicine agencies have given some approval for some of the organ on chip for the testing, for toxicity studies and all, but still the exact regulatory agencies and the exact approval from these regulatory agencies is required, and it is an ongoing process where they can develop their own guidelines for the best possible use of organ on chip technologies for the drug testing and drug discovery. Next to that, there is a technical expertise. The limitation is technical expertise, that all the people are not that much aware about the utilization of the organ on chip technology. They are not much more aware about how it can be developed, how much amount or how much money is required, how much time is required, and what will happen if there is a failure of the development of the organ on chip technology. So in those circumstances, a need for the specialized training in microfluidics, cell culture and data interpretation is very, very important. So these are the current limitations, like standardization and reproducibility, scalability and manufacturing, regulatory approval and technical expertise. In case of infectious diseases research, as the major problem was taken place in COVID-19, and similarly, many of the virus or bacteria or microorganisms may attack the respiratory system. So in those circumstances, the lung on chip can be used to study how the viruses and like COVID-19 or any other type of positive agents infect the lung cells and test the antiviral drugs. So not only antiviral drugs, even the antibiotics can be checked, antibacterial can be checked on the lung on chip, which is specifically causing the respiratory complications. Similarly, intestine on chip, which is helpful for to develop the model gut infections, where chronic infections like inflammatory bowel diseases and chronic infection in the GIT, where the antibiotics become resistant and they are not that much effective. For that, if we are using a drug for such a chronic condition, how long that drug should be utilized so that it cannot develop the resistance, or how the combination can be utilized so to prevent the resistance, or once the resistance occur, a new molecule, how it can be utilized and implemented for the treatment of such a chronic disease that can be identified with the intestine on a chip. For example, Fish Institute used the lung chips to test COVID-19 treatment faster than the traditional method. So it is one of the example where the Fish Institute, they identified the lung on chip for the assessment of effectiveness of drug for the management of COVID-19 infection. Similarly, in case of neurological and neurodegenerative disorders, where the blood-brain barrier on chip is one of the important tool which can be used as a model that how the drugs cross the brain protective barrier, that is blood-brain barrier, aiding the developing treatments for Alzheimer's disease, multiple sclerosis, and even the brain tumor. Because though most of the drugs are having very good anti-cancer activity or very good we can say neuroprotective activity or potential, but still the chances are there they cannot cross the blood-brain barrier upon the administration. So in those circumstances, how best the permeability through the blood-brain barrier can be improved, that can be analyzed using this blood-brain barrier on chip technology, where the Michael J. Fox Foundation supports the organ-on-chip research for the Parkinson disease therapies, where specifically in case of Parkinson disease, the drug which will be used and generally the dopamine level is going to be affected. To enhance the dopamine level, some stimulants or some precursors can be given like levodopa. But chances are there the peripheral metabolism or some limitations of a particular drug is unable to cross the blood-brain barrier and it cannot show the activity. So for that purpose, such type of organ-on-chip technology helps to improve the permeability, helps to identify the exact outcome, and helps to identify even the adverse outcome also. Similarly, in case of the diabetes and metabolic disorders, where the pancreas on chip, liver on chip can be identified. It helps to in the diabetes research by stimulating the insulin production and testing a new treatments. Along with that, in case of liver on chip technology, where it aids to understanding the fatty liver disease and metabolic disorders. Most of the time that fatty liver disease occur because of the diabetes like condition, hypertension, along with that certain habits like smoking and alcohol. So those kinds of things which will make the patient aware that, see, if you are continuing the such type of alcoholism and smoking like condition, your liver will get affected and there might be a fatty liver. That can be predicted with the help of liver on chip, when it is coming in contact or when it is exposed with the similar kind of concentrations of alcohol and nicotine like substances and even any drug or drug metabolites also. So it can give the predictions for the toxicities occur to the particular organ. Scientists at MIT and Harvard University are using pancreatic chips to develop more effective diabetes treatment. So definitely that how the drug which are going to stimulate the insulin secretion that can be identified. It is one of the important mechanism for the management of the diabetes. So these kinds of chips will be more useful to assess the drug utilization and drug effectiveness. This organ-on-chip technology reducing the animal testing and ethical advancement. So that's why it favors the ethical committee that if we are going to use the advanced technology like organ-on-chip, it will regulatory agencies like FDA, the European Medicine Agency, and NIH, that is National Institutes of Healthcare, are actively promoting the organ-on-chip technologies as an alternative to the animal testing. So if it will become alternative to animal testing, definitely usage of animal will get reduced and ethical committee will support to such type of advanced technology. The FDA's Modernization Act 2.0 passed in 2022, which allows the drug companies to use the organ-on-chip technology data instead of animal testing for the preclinical research, specifically for the toxicity data and the efficacy. So this kind of advancement and support from the regulatory authority helpful to enhance the effectiveness and utilization of these novel utility tools. In case of future healthcare, how this organ-on-chip technology will be useful, specifically that organ transplantation, that organ-on-chip technology could help the test donor-recipient compatibility. Along with that, in case of aging research, where simulating how aging affects the drug metabolism and disease progression, and specifically in case of space medicine, where NASA uses organ-on-chip technology to study the effects of microgravity on the human organ. So these are the future perspectives where the organ-on-chip technology can be developed and can be identified and can be utilized. Along with that, there are the major limitations which we can see, that the cost effectiveness, advanced facilities and requirements, expert people from the different domain, and the collaborative work. It is a collaborative work, so there are a number of domains need to be worked together, and this collaboration will little bit take time, and it is time consuming, as well as we should get the approval from all the domains so that they are agree to work together to develop such type of organ-on-chip technology for the further development of the drug and analysis of the drug for their therapeutic as well as toxic effect. Finally, ethical concern will be there in case of personalized medication, where organ-on-chip technology will be utilized, where from the patient some cells, some tissue need to be extracted. So it requires some ethical approval also. So the ethical concern also there, it is considered as one of the limitation of utilization of this particular technology. But there is a hope, as we have seen that traditionally that how the transformation is taken place in front of us only. We are seeing that how the old antenna mobile was converted to the keypad mobile, and later on the smartphone come into the market. So similarly, this is also one of the advanced tool, advanced novel technology, and the people will accept and adopt this technology based upon the research which is going on and the regulatory authority which approve the research on the particular field. Coming to the conclusion, organ-on-chip technology is transforming pharmacology education and research by offering a human-relevant, efficient platform for studying the drug effects and diseases. As the technology evolves, it holds the potential to bridge the gap between lab-based research and the clinical application, ultimately improving the drug development and the patient care, which is a need of time. Advanced organ-on-chip research is pushing the boundaries of drug discovery, disease modeling, and personalized medicine by integrating the AI, integrating the artificial intelligence, the stem cells technology, and multiple organ system along with the machine learning. Organ-on-chip technologies have the potential to transform the biomedical research into clinical application in the coming years. With the continued development, organ-on-chip technology could soon be a standard tool in hospitals, research lab, and pharmaceutical industries, leading to a faster, safer, and more effective healthcare solutions. So with this, I want to conclude my topic on organ-on-chip, which is one of the advanced and which can give the real-time effect of the drug for during its development and testing its toxicity and the therapeutic outcomes. Thank you. So the session is open for the question.
Yes. Sir, there is a question from Pranita. Organ-on-chip technology, how it is cost-effective?
Organ-on-chip technology nowadays, as it is in emerging conditions, so it is not fully developed. So that's why what are the material, raw material, or we can say that the the tools which we are using or advanced techniques we are using, it is little bit costly, and the facility also not available in our routine labs, or we can say that the place like developing countries, it is very difficult to develop the organ on chip. So advanced technologies, expertise from the different domain are very important, and that's why it will little bit expensive in present condition. But in coming future, definitely as the advancement will be there, the research will get explored, that it will be cost effective and it will be very useful also.
The second question is from Dr. Umair Ahmed Nehal Ahmed. How much effective is the organ on chip technology for cancer treatment?
Yeah, it will be effective because cell proliferation, the rate of cell proliferation can be identified in vitro. At the same time, effectiveness of the toxicity or toxic impact of the drug on the particular cancerous cell can be identified. Along with that, the treatment will be more specific for with the help of organ on chip, that how we can enhance the therapeutic outcome of anti-cancer drug and how we can minimize the adverse outcome of the anti-cancer drug on different organ can be identified. That's why it will be more useful for the treatment of cancer. The concentration, even the concentration, how much concentration required to kill the cancerous cell, that we can identify in vitro.
There are no more questions, sir.
Okay.
On behalf of Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, I would like to extend a sincere thank you to Dr. Ketan Hatware, sir, for such an insightful and inspiring session. So, as a token of appreciation, please accept this certificate.
Yeah, thank you very much, sir, for giving me this opportunity to give some brief talk on the novel technology which can be useful for new drug development and toxicity studies. Thank you.
Definitely, definitely. Thank you. Thank you so
Thank you.
much, sir. Thanks
Thank
for.
you.